Good morning once again and welcome to CIET NCERT live interaction program. This is Dharmendra Singh and now what we have, it's a very interesting topic to be discussed about, and what is that? That is hereditary and evolution part second for the students of class 10, of course from the science. And the expert we have got to explain all the things very easily so a student can learn it and can remember for a long time. I would like to welcome Dr. C.B. Shumre, Associate Professor, Department of Education and Mathematics. I welcome you, ma'am. So what you have today, before we talk about this, I need to give you more information. If you have any questions, any query, you can get back to us on our toll-free number, which is 1-800-111-265. 1-800-112-199. Apart of this, if you have any questions, if you would like to send through email, we do have our official email ID, that is ciet.kishoremanch@gmail.com. Top of that, if you would like to catch us with the live session, you need to go to the NCERT official YouTube channel and there would be a comment section. You can put your questions over there and we will try our best to take few of those and discuss with the expert. Apart of this, this program can be viewed on some television subscribers as well, like SWAYAM Prabha, that is on channel number 3131, DD Fresh Dish TV, that is on number 128, and you can see this program as well on TataSky subscribers, that is 756, and of course Airtel TV 440. So these are the basic information where you can get back to us. Now I would like to go to our expert and try to know what we have today, something mysterious. And ma'am, I would like to request you if we can recap some things with the previous sessions, so we can go ahead.
Sure. Okay, thank you. So yes, we had part 1 of this chapter last week, so let me just go back and discuss a few things. We're not going to recap all of it, but we are going to recap just a few of it. So let me just take you to some of the slides. To start with, I have a slide about Darwin, which is also in the NCERT textbook. So we discussed a few things about Mendel, Gregor Johann Mendel. So who is Gregor Johann Mendel? So he is also known as the father of modern genetics. So we discussed a few things about how he went about his experiments, how he did the experiments, why he used a pea plant, and how he used his knowledge of science and mathematics together so as to come out with all the laws that we know about it today. So he did a lot of crosses. I mean, he used a pea plant, and last week we discussed a few things about why he chose pea plant. So he chose pea plant because of certain characteristics that he found. For example, there were some I have that slide for you, where there were very clear contrasting characters. So he found 7 of those contrasting characters. For example, he had those flower color. So it was totally clear that some pea plants were bearing purple color flower. Some pea plants were bearing white color flower, and the position of the flowers. Some pea plants had clearly axial, that means not at the tip. Then some of those pea plants had terminal flowers. Then the seed color. Some of the pea plants had yellow seed color, and some of those others have the green. Then similarly, he also found contrasting characters in the seed shape, the pod shape, and the pod color, and the stem length, that is how tall or how short the plant is. So these contrasting characters were very helpful for him in order to cross them, because if you're clear about what you have, then it is easier for him to also predict. And as I said, he had knowledge of science and mathematics, and so he utilized those knowledge to the maximum possible, because of which he was able to predict few things, come up with some kind of ratios, which today we know the laws of inheritance. So that's how Mendel went about cultivating pea plant using those different characters. So he came up with different laws, as I said. So then what did he do? So he did some crosses. For example, the monohybrid cross. We discussed a few things about monohybrid cross last time. So what is this monohybrid cross? A cross between individuals heterozygous for one character or a trait. A cross between individuals heterozygous for one trait or one character. So last time again we studied about what these heterozygous individuals are. So if I just take you back to the previous slide, for example this slide. So this slide is an example where it shows we have a homologous chromosome there, a homologous chromosome with heterozygous genes there. The alleles are different. For example, you can see in the picture, it has in the upper chromosome it has the allele for purple color. In the lower chromosome it has allele for white flower color. So this is a heterozygote, or this plant is a heterozygous individual because it has the two versions of the alleles. So that's about, so in a monohybrid cross, you cross individuals which have different alleles for the same character. So that's what he did for the monohybrid cross. So if we go back to that, similarly he did some crosses for the dihybrid cross. That means that he took two characters. Again, in the individuals are heterozygous for two characters. That means that it could be any of those two contrasting characters that we just discussed. So he did all kinds of crosses, all kinds of hybridization. So using different characters, crossing between different characters, as we saw the different contrasting characters. So that's what he did. So let me go back to the I think maybe we can recap the monohybrid cross. Yes. Last time we discussed a few things about how he went about the monohybrid cross and what he got, or what he conclude from the monohybrid crossing. So to start with, if I can take you to this slide where the monohybrid cross is happening. So you can see that slide. So this is a monohybrid cross. So to have a monohybrid, that means to have an individual which have the different versions of this of the allele for the same character. For example, the previous slide I showed you about the homologous chromosome where it had two different versions for the same character. That means one chromosome had allele for the purple color flower, another chromosome had allele for the white flower color. So in order to get individual like that, what he had to do, or what we have to do, is we have to take pure breed or true breed plants, true breed or the pure breed parents or the plants. So we discussed again last time what are those true breeds or the pure breeds. So true breeds or the pure breed plants will produce the same kind of phenotype generation after generation. And so how do you obtain that? You obtain that by selecting it. So you choose some of those characters that you want, you self-fertilize that again and again over the generations, and you'll ultimately find that when you grow that same plant again and again, self-fertilize it again and again, you'll find you'll have the same phenotypic character in the following generation. So that's about the true breed or the pure breed parents or the flowers or the plants. So what he did was he took two plants, as in the slide. We can go back to the slide there. So he took these two plants, two pure breed plants. That means one plant, pure breed, which is pure bred for purple flower color. That means that one plant, the plant on the left hand side with purple color, this, if you grow this plant, it will produce individuals which have, or plants which have all purple flower color. On the right hand side, he took another plant parent which bears the white color, which is a true breed. Again, that means that if you grow this white flower color and if you self-fertilize this or self-pollinate this, then it will give rise only to white flower color. And so when he crossed these two plants, so when I say when he crossed these two plants, it's not just crossing the plants and the plants just fuse and they come up with a new individual. It doesn't happen that way. We know about pollination. We have learned about how pollination happens. So pollination, the process of pollination, the pollen from one plant is transferred to the another plant. So in this case, you can either transfer the pollen from the purple flower or the pollen from the white flower to the other. So either way it's fine, because in plants you know that it's going to be the same. So he pollinated that way. He took these two pure breed plants and he pollinated between this. Then this, after pollination, what will happen is, after pollination, hybridization happens. After hybridization, then you find fruits or the seeds that produces. So after the seeds are produced, then he grew those seeds, and when he grew those seeds, what he found was all those seeds, the plants that coming out from those seeds bear all purple color flower. So now he found that it is all purple color. But in the parents, he had taken one of the parents were white bearing white color flowers, whereas the other one was bearing purple color flower. But in this case, after hybridization, what he found was that the seeds that were produced from it, when he grew them, he found that all the plants that came up from those seeds, they bore purple color flower. So when he self-pollinate this, now this is a hybrid, because it is produced from the hybridization of two kinds of flowers, plants bearing purple color flower and plants bearing white flowers. So now the hybrid is a purple flower color phenotypically. That is what he observed, or that is what we can see. And so when you pollinate, self-hybridize these kinds of plants, that means plants in this F1 generation, as you just saw in the slide. So the F1 generation, or the individuals that came out from the hybridization of those two kinds of flowers, now they are hybrid, you know. So they are hybrid. When you hybridize these kinds of plants, what he found was that in the next generation, again, when he hybridized these two, that means that it is about, again, pollination happening there. So either way, because this is the same kind of plants that is found in the first generation, so he can self-hybridize or he can cross-hybridize. It's going to be the same. So whatever he does, you're going to get the same thing. So either you go for self-pollination or you go for cross-pollination. But since we are using the same type of flower, so the result is going to be same. So for what happens after that, after pollination happens in those kinds of flowers bearing all purple color flower. So he found that the next generation, the plants coming out from the seeds after crossing those flowers, he found that majority of them had purple color flowers, but some of them had white color flowers. So that is interesting. So in the parent, initially what we had was two forms of two pure breeds, one for the purple color, one for the white color. Then in the next generation, when the offsprings were formed, we found that all the plants bore purple color flower. But when you self-hybridize or when you cross-hybridize these kinds of hybrids which bore the purple color flower, now you find that in the next generation you find that they have, some of the plants had purple color flowers, some of the plants had white color flowers. That means that the white color that we initially had reappeared in this generation, you know, in the second generation. So that's what he found. And so if we look at the numbers, when he looked at the numbers, for example, out of those more than 900 plants that he counted, he found that 705 plants bore purple color flower and 224 plants bore the white flower color. So if you calculate ratio wise, approximately it comes out to be like 3:1. That means 705 is to 224, it gives you roughly 3:1 ratio. That means you have majority of those purple color flowers, you have some with the white flower color. That means the ratio is 3:1. So he did similar kinds of hybridization. I mean, he took different characters again. So this was just one example where he took purple, the flower color. He took another contrasting, another character where it could be seed shape or the seed pod, things like that. So, but the results were almost the same. For example, before I get into that, I'll just show you. So that the previous slide was about phenotypically. So what was observed. But if we get into the genes, so what is exactly happening in the genes? Why are we seeing the purple color flower in one generation? In the next generation, why are we seeing the white color flower again? So if we look at the genes of those things, so we can go back to the slide there. If we can go to the slide. So you can see there. So the parent generation again, the true breed. So genetically, if we speak, so when we say it is a true breed, that means that it is homozygous for that character. Homozygous. So what is homozygous? That means that it has the same allele. The same allele. For example, in the purple flower color on the left hand side, you'll find that it is both, it is homozygous. It is the purple color, again the dominant one. So we previous session again we discussed about what dominant is, what recessive characters are. Dominant characters are those which are expressed in the heterozygous condition. For example, if a homologous chromosome has two versions of the allele, a purple allele for the purple color and allele for the white color, then you will find that the plant will always bear the purple flower color because it is dominant over the white flower. So that's what. So genetically, if you speak in this slide, the parent generation, the true breed, that is, it will be homozygous. Homozygous in the sense that you can see the big capital P's over there. I don't know if you are able to see that. So on the top, on the left hand side, just below the purple flower color, the big two capital P's indicate that they are dominant and they are homozygous for the character. That is the same big P, homozygous, and they are the dominant. Whereas on the right hand side, you find the small two P's, that is, they are homozygous because they are same, the alleles are same, but now they are recessive. That is why we represent them as small P's. Am I clear about that? Okay. So when you fertilize these two plants with with the homozygous recessive character and the homozygous dominant characters, these two plants, when you hybridize them, what you find is the hybrid that you find has purple flower color, but the genetic constitution is that it is now heterozygous. So we can just go back to the slide to show what that heterozygous is. So you can see there in the F1 generation, it is a hybrid now. It is a purple flower color. All the plants will bear the purple flower color, but now the genetic constitution is big P and small p, that is, they are heterozygous. Whereas in the parent generation, they were homozygous. Either they were two big P's or they were two small P's. But in the F1 generation, now all the plants had the genetic constitution of big P and small p, that is, they are heterozygous now. So when you cross hybridize these two or self hybridize these kinds of flowers, what you find in the next generation is it will bear fruits. Then the fruits, when you grow them, you will find that again similarly you'll find the purple flower color coming up and the white flower plants coming up in the next generation. But how that is happening, you can see in the slide on the right hand side of the slide. I have just enlarged the picture on the left hand side, so it's the same thing that you are seeing on the bottom of the left hand side. I have just enlarged it on the right hand side, so you can see there. So you can see if plant has a genetic constitution which is heterozygous, that is big P and small p, then what are the kinds of gametes it can produce? So we know that a gamete can have only one of those P's. It cannot have both the P's. We studied about what a gamete is. What a gamete has haploid cells, So it cannot have both. So it gets one of those allele from one parent. It gets one of those allele from another parent, and that's how they get two of those. So what kind of gametes this heterozygous flower or a hybrid that is formed in the F1 generation, what kind of gametes they can form is, they can form two kinds of gametes. As you can see in the slide on the right hand side, big P and small p. That is, they can only form two kinds of gametes. And similarly, you can take either, because you always need two parents. So you can either take one on the left hand side, on the top or the bottom. You can take one as egg representing egg and the another representing sperm. So that's how the zygote is formed in the process after the process of fertilization. So similarly, another plant can form gametes of big P and small p. The kinds of gametes that they can form are same because the parents are same. The parents have same genetic constitution. That means they are heterozygous for that flower color now. So since the parents have big P and the small p, that is, they are heterozygous for that flower color, the kind of gametes they can form is big P and the small p, both, whether it's a sperm or the egg. So that's what they're going to form. So when you produce individuals out of this, so you can go back to the slide again. So when you hybridize this, what's happening is, so on the top there is a big P there and there is a small p there. Then on the left hand side, on the right hand side of the slide, but on the left hand side of the table, you can see the eggs form. X from F1, that is again the big P and the small p. So if you cross them, you can see there, first individual on the first corner, it forms two big P's. That means now they are homozygous individuals are produced. The flower is homozygous for the flower character, and it is dominant. That is because it is big P, and that is the reason you have purple color flower. Then the next individual that you can find on the right hand side, again on the top of the table, you have one big P and one small p. That is, they are heterozygous now. But still, since you have the big P, which is a dominant form of the allele, so you will still find that plant bearing purple color flower. Come down to the next individual that is formed from the cross of big P on the top and small p on the bottom of the right hand side of the table. You'll find that the individual form is again big P and small p. So what kind of individuals they are from? Again, they are heterozygous because they have one big P and one small p, but the kind of flowers they are going to produce is, it's going to be purple again because it has the dominant allele there, the big P. But the last one, if you see there, you see the white flower color. So what is the genetic constitution of that white flower color? It is two small p's there. So two small p's, so there is no big P there, and so when the recessive allele are present in the homozygous condition, when the recessive alleles are present in the homozygous condition, you find that character is expressed. So it is only when two of those recessive alleles are present in the plant, then you come across, then you get this white flower color. Otherwise, as long as there is the dominant allele, whether in twos, both the alleles are two dominant alleles or single dominant alleles, as long as that dominant allele is present, you will find the dominant character expressed phenotypically. But as you can see in the slide, the genes where the genetic constitution were different, and so we still say that, because you can see that the kinds of individuals that they can produce are, in this slide, 3 flowers with purple color and 1 flower with white color. That is 3:1 ratio. But if you look, that is a phenotypic ratio. That is what is seen, what is visible. But if you look at the genetic constitution, which we cannot see looking at the flowers, but if you look at the genes, you can see that, for example, the first box, it has 2 big P's. That means 1. Just count that 1. And the second box has 1 big P and small big p, heterozygous condition. And the third one also has 1 big P and 1 small p. They are also in heterozygous condition. That means that 2 similar kinds of genetic constitution we can find now. Then the fourth one is 2 small p's. So genetically, if we speak, we find that there are 3 varieties there. One bearing 2 big P's, that means bearing both the dominant allele. 2 plants bearing heterozygous condition, that means 1 dominant allele and 1 recessive allele. So still the dominant allele is going to be expressed. That's why you still have the purple flower color. But the fourth one, you have 2 recessive allele, and so you have the white flower color. So phenotypically it is 3:1 ratio, but genotypically we say that it is 1:2:1 ratio. So that's the difference. So although the phenotypes are expressions of the genes, of the genotypes, it doesn't say everything about what is there in the genes. It doesn't say everything about the genetic constitution. So we really need to understand the genetic constitution so as to know what is going to happen in the next generation again. So that's what he did. So what he did, that was just one example of flower color. He did for all kinds of characters, contrasting characters. So you can see there in the slide. So if you go to that slide, you can see there in the slide, you can see the ratio there. So for the flower color, as I said, 705:224 phenotypic ratio. That means it's approximately coming up to 3.15:1 ratio. So if you round that up, it is like 3:1 ratio. Similarly, for another contrasting character, when he crossed those 2 characters, for example, the flower position, he found that of those 800 plus flowers, he found that 651 flowers bore the axial, then 207 flowers bore the terminal kinds of flowers. So again, roughly coming up to 3 1:1 ratio. So all those contrasting characters, when he bred them, when he crossed them, the true breed, initially starting with the true breed form, and the hybrids when he crossed them, this is the ratio that he found. So that's what happened during this crosses of the monohybrids. That means the hybrids with heterozygous alleles. So, and then what happened? So what are his findings, what are his conclusions from those crosses after he studied the monohybrid cross? So we can just go to the slide there. So what did he find? Findings are like, first finding was alternate versions or the alleles of genes account for variation in inherited characters. That means the inherited characters differ because they had alternate versions. For example, in this case, the case that we saw, the two versions were the purple flower color and the white flower color. So that's what he concluded. Second one, he concluded that for each character, an organism inherits 2 copies of a gene, one from each parent. So as I said, if the hybrid is heterozygous, then the gamete that can be formed is only either the big P or the small p, and when they fuse again, so one of those will go to then it will take another gamete from another parent. So when these 2 gametes hybridize or fertilize, then you get the zygote. So yeah, so going on to that, so that's how it happens. So you find that every individual inherits one allele from one parent, another allele from another parent. The third thing he found was, if 2 alleles at a locus differ, for example, definitely in the slide that I showed you about the homologous chromosome, where the upper chromosome had allele for the purple color and the lower chromosome had the allele for the white flower color. In such cases, one of those characters will be expressed, one of those characters will be seen. That means in this case we found that purple flower colors are always expressed even though the recessive allele is present. So that's what he found. Then the fourth thing is that he found that 2 alleles for a heritable character segregate during gamete formation. That means if a parent is heterozygous, for example, it had purple flower color allele as well as white flower color allele. So even if it has that, okay, so there's some issues with the lighting system here, power here, and so we are having some trouble here.
You continue, ma'am.
But we are going to continue with that. So the fourth and most important conclusion was that the 2 alleles for a heritable character segregate during gamete formation. That means that if an individual or a plant is heterozygous for a character, for example in the hybrid that we saw, which is heterozygous for the flower color, which had big P and the small p, these 2 alleles are going to segregate during the formation of gametes. That means both of these alleles cannot go into one of the gamete. So it has to always segregate. Only one of those allele will go to one gamete and another allele will go to another gamete. That's how. So there is segregation of those alleles during the formation of the gamete. So that's what he found from his monohybrid crosses.
Right.
Yeah, that's a really lengthy explanation out there.
It was lengthy, but it was very interesting, and the way you conclude all this, I think students should get this in a easy way, and when they will go to the exam center, they will explain in a respectively manner. There is some question I think I would like to raise here. I think that is in your order or not, but you can take it.
Yep.
The question is, how is biological sex of human beings determined?
Okay, so in that case, I think I have a slide, but I think are the slides working? Okay, so I can show you this slide. So sex determination is something which is very controversial also. But genetically, as a science student, it should not be controversial. We are just trying to understand how a sex is determined in an individual. So as you can see in that slide there, so what happens in human beings, in plants, earlier I said that it's all the same. whether you take one plant or another, so it's going to be the same. But in human beings we have something called, that's not in all the plants but in most of the plants that happens, but in human beings it's very clear we have male and female. And so the gametes are also different. We have studied about, students have studied about the gametes, male gametes which are sperms, female gametes which are eggs or the ovum. So those are the gametes. So how are these gametes released, or how are these gametes fertilized, finally forming the zygote and finally forming the individual new offspring? So in human beings, as we all know, the possible gamete is we call it in males, it is a sperm which carries one Y chromosome, and or it can carry one X chromosome, one of those. But it is not just one X and one Y. When we say that it is a gamete, it has another 22 autosomes plus one sex chromosome in a gamete. So a male gamete, that is a sperm, can have 22 autosomes, which they have studied, we have explained this in the previous session. So a male gamete, that is a sperm, will have 22 autosomes plus one sex chromosome, which can be either X or Y. Whereas a female gamete, which is called egg or ovum, can have 22 autosomes, and it will be always X chromosome. So X is always there for the egg or the ovum for female gametes, but for the male gametes it can be either Y or the X chromosome there. So when these two fuse, what happens is students are aware about fertilization. So during fertilization, either of this male sperm which carries either X chromosome or the Y chromosome, they will fertilize with the eggs, the egg or the ovum. So it is chance. Nobody is responsible for anything, because we try to fix responsibility whether female is responsible for the sex of an individual or the male is responsible for the sex of the individual. So we try to fix responsibility, which is not true. It is by chance that the sperm with X chromosome or the sperm with Y chromosome fertilizes with the egg or the female gamete. So it is by chance. So in case the sperm with X chromosome fertilizes with the egg, then it will, the individual that's going to be formed is a female, because now they're going to have two X's there. But if the sperm that is fertilizing with the female has Y chromosome in it, then it is going to be the individual that is going to produce out of those fertilization, it's going to be male, because it has one X and one Y. So it is by chance. It is all about chance, and nobody is responsible for that, which has become very controversial. And again, sex determination is something which is really concerning for a country like us, because we are aware about the social evils that's happening, where female feticide that's happening over there, trying to kill the fetus if they find that the female is there. So the dislike for the choice for male child, things are happening out there, and we abuse also the medical diagnosis techniques that's available for other kinds of treatments. For example, amniocentesis, which is meant to determine the genetic disorders so that a healthy child can be produced. So but we are misusing that. We also misuse ultrasounds and all to determine the sex, so that we do what we want with the fetus, and so many illegal things are also happening out there. And so sex determination is something very controversial, and it is going to impact the society as such. There can be a lot of conflict, because if there is a population imbalance of male and female, what can happen, you cannot even imagine. So things are beyond just the sex. Yes, as science students we are concerned about how a sex is determined, how a female child is born or a male child is born, but beyond that, students also need to think about what exactly happens in the society when a male child is. We heard about beti bachao, beti badhao.
Yes.
It is all because of that, because of the things, evil things that's happening in the some of the increasingly people are getting aware, and importance of female child is felt more increasingly. Now people are feeling that, okay, without girls or without female, how the society is going to run, or how the society is going to function, So things are, people are aware, and awareness, a lot of awareness program is happening there. So it is something good. So students also need to think beyond the sex and think about the impact of the sex in the society. So as a student, since they are going to be part of the society and they are part of the society, it's important for them to sometimes introspect into all these social things that's happening related to sex.
Right.
Yeah.
which is not questionable, and you need to force for this. I think the way you explained all the things, thank you so much, ma'am, because we are lacking with the time and I wish to continue this, but we are very much bounded. So I believe the student must have got all the lessons the way our expert has explained all the things with the conclusion. So you need to follow the same thing, and for the practice, do you have any tips to give them for practice, how they can remember all the things in a one go?
Yes, they can go back, they can just because we have crossed some of those things, they can take some other characteristics. For example, if they did not take the pure breed, for example, we started with the pure breed there in the slide. So if they did not take this pure breed, then what happens? So they can just try crossing those. Initially, for example, in that slide you can see there. So if they had not taken the pure breed, or they had not taken the homozygous condition, if they had taken one big P and one small p and cross it with two small p's, what happens? So they can just play around with those kinds of crosses and see what happens in the next generation. So that will give them a good idea about how exactly characters are going to be inherited in the next generation. So that's something they can practice.
There you are. So you got the lesson, you got the motive, you got the tips, how you can increase your knowledge. So thank you so much, ma'am, for being with us, for this lovely information you provide to our students, and including me also. And you just keep watching Kishore Manch, because after this we would have a lot more exciting topics to be discussed. So don't go anywhere. Keep watching Kishore Manch and let us allow to go. Thank you so much.